So our group did great. This is going to be kind of repetitious because they were really good. And we're just going to go through the answers really fast to make sure that everybody got them. And let me, oh, there we go. I have to. There, I got it there. So how many people enrolled? Well, there were 32 screened, 23 enrolled, but only 10 were treated and 13 were the control population. And as the point we'll make in a few minutes is those 13 were the people who met entry criteria, but said, I don't want the surgery. So it was not randomized. How long was the trial? It went a year. Was there a sugar pill or sham surgery used? No. Was the study blinded? In other words, did the investigator know who was treated and who wasn't? The study was not blinded. So we're hearing a lot of red flags, right? And now you would recognize those red flags if you read it after listening to Dr. Macklin and after my talk about how to read a paper, I hope. And how did they measure safety and how did they measure efficacy? And this was in the methods and data section. Well, there were no plans for safety. They didn't outline, we're going to follow X, Y, and Z to make sure that this treatment is safe. For efficacy, they looked at survival. They looked at the functional rating score, FVC, and quality of life. What were the inclusion and exclusion criteria? Well, the inclusion were listed: confirmed ALS by Ellis Scorial with neurophysiology, EMG, MRIs that didn't show anything that could explain the findings. And then it's not clear, but they could do an FVC is the best I could tell that meant, and an appropriate nutritional state. And the exclusion criteria were severe bulbar involvement, inadequate nutritional state, whatever that meant, tracheostomy or gastrostomy, the presence of neurologic or systemic disorders, and some problem on MRI that could explain findings. So now, how were the treatment and control groups chosen? Well, it was done by the site investigator. It was not random, and the controls were self-selected. They just said, I don't want that surgery. So, you know, does anybody think this is a good design? Yeah, everybody who I can see is shaking their head no, and that's the right answer. So how do we compare the measures between the control and treatment groups, right? You want them to look the same, you know, because if I'm comparing disease, I want the disease to be similar in the two groups. So let's start with duration of ALS diagnosis to baseline visit. You know, when were they diagnosed and when was their baseline visit? Were those the same? And again, as I mentioned, looking at tables and graphs are often, the figures give you a lot. And here's our table. And here you can see the duration in the control versus the treatment group. And those kind of look a little bit different, don't they? So, you know, the treatment group was 30 months compared to the control group at 14 months. And in our group, we talked a little bit about that. Certainly they're different. Either the treatment group was further along in their disease. That's one possibility, which would make it pretty bad control group, or they were slower progressors. Again, something that would make it not a great design. And then the ALSFRS score at baseline, again, if we look at table 1, here's our ALSFRS scores, and it looks like the controls are a little bit higher than the group. And here you can see at baseline, the control group had a higher score, the ALSFRS score, than the treatment group. And over time, I would argue that this shows that the control group were more rapidly progressing, and that now the treated group looks like they were slower, but I don't know whether that's because they were slower progressors to begin with or the treatment worked. So that the ALSFRS-R score was very different. And so are these treatment and control groups similar? Can they be compared? That's the real question. Can they be compared? And the answer is, of course, they're very different. They're apples and oranges. What percentage of people died in that 1 month after surgery? 1 out of 10, which is a 10% death rate. So how can we improve this, right? What would be better? How can we do it better? Well, you could have it placebo controlled, although, you know, as Eric commented in our group, well, you know, sham surgery is not the greatest thing, and maybe there would be a better way to do things. And Eric, you can comment on that if you'd like. Blinding, you'd want somebody who is blind to whether or not the person had surgery and stem cell treatment or not, doing the assessments. Randomization, an important thing for the confounding. Those groups were very different. If you took, as you know, Dr. Mackman was pointing out in our group, and he can comment, if you took only those folks who said, I want the surgery, and then you randomized them to surgery or not, that would have been a much better control versus treatment group. You should have been collecting safety measures. You know, that certainly would have been important, and adverse events. And, you know, you want to know whether or not there were any protocol deviations. Did they follow the protocol they set out to follow in terms of timing, treatment? You know, that would all be important. So anyone have any questions or comments on all of this? Question? No questions. Just unmute and yell it out or put it in the box. So final lessons, read carefully. Don't take everything at face value, right? You thought this is published in a medical journal. It must be right. These guys must be smart. This must be good stuff. I want to go out and get stem cells. Please put a hole in my head and throw some stem cells in it, right? But now you know how to read carefully and really be more critical, be informed, and educate others. That's your job now. Thanks for being a research ambassador. We really appreciate it. I'm going to stop my share. And any comments or questions? I think we have something in the chat.
I do have a question. Is there a way I could it's being recorded, is there a way I could rewatch any of this at like a certain place?
Yeah, Phil, it's going to all be posted. We're recording everything. It'll be posted on our website and with links, probably on the YouTube channel. Sarah, where are you going to post this?
Yeah, we're gonna have them all uploaded to YouTube, and then I'll share the links with everyone.
But yeah, the link should also be on our website.
Mm-hmm.
Thank you.
Yeah, Phil, no, please. We're glad you're here, and you know.
So Terry, I guess I might share what I shared in our group, that I feel like there's a balance here. These are the first 10 patients, presumably, who were treated in this way at this one clinic. There's a benefit to that group publishing these results. I don't agree with all of the ways that they chose for presentation, but they went to substantial effort to collect a lot of data, to document it, to put it in the literature. That gives us a basis for the next set of studies to see whether stem cell therapy might be beneficial. Just as Terry's outlined, there's some serious deficits in the current study, but and thinking about how to improve it is exactly the right direction. But I wouldn't throw out all these results because there are some obvious failings. It's the right place to start. you wouldn't necessarily you wouldn't appropriately start with a 500-person double-blind, sham surgery trial for something as invasive as stem cell therapy. So I don't want to undercut all of the effort that the researchers went to and sort of position this in a sequence of exploration. So yes, clearly the lack of randomization, I think, is sort of a fundamental failing here. It's we're really unable, to my mind, to make a judgment about whether the differences in the two groups were due to confounding because of the obvious differences at baseline, or whether that they were potentially due to some efficacy from the stem cells. And it wouldn't have been too difficult to provide some randomization, but that wasn't the data they had in hand. At least they did provide some comparator data, just not very good comparator data.
Perhaps it would be best to consider this almost like a phase 1 safety and not present it as efficacy.
Right.
So this was this safe 10% surgical loss. That might have been a better way to present it. That they seem to be pretty stable, but this was not powered for efficacy, especially since the control group was not an appropriate control. It wasn't a matched control group. Right. I don't know if anybody else has anything to add to that.
I'll just echo what Eric said. I mean, I applaud anybody who's trying to do ALS research. It's not easy, but I mean, they clearly set up a study. It had oversight. They were measuring things. They published it for peer review. That sets them way above a lot of other people who are out there just advertising treatments without doing all this. But I mean, the two big problems were this should have been mainly a safety study, and there isn't even a table of adverse events. I don't know how they got away without creating a table of adverse events, because we don't know if we can move this to the next level without that. We don't know exactly how safe this was. And then the other is every study has limitations. I've never read a perfect study.
Right.
But ideally, you would actually put in your discussion section, hey, this is what we did. This is what we found. We know that there's these limitations, and here's some suggestions for improvement. And they just didn't recognize their own limitations. That's, to me, a problem.
Rick, I agree with you. You mentioned the peer review process, and I also agree with Eric. I applaud everybody who shares the data. Where were the reviewers here also? Right. I think that peer review is a very healthy process, precisely because the reviewers are doing the exercise, should be doing the exercise that we do here. Not to prevent anything to be shared and anything to be published, but to prevent really what may happen, to give really the patient community and everybody else false hope, right? As Terry said, people may think, oh, this was published in a very good medical journal, and therefore it must be true. So I really think that we also in the community should be doing a better job in peer reviewing one another and to avoid this.
Yeah, Matt has a comment, where should we be looking? Where in the literature might we find reputable studies on stem cell therapy? And there's a broad range, there's thousands of medical journals out there, broad range. This cytotherapy one is very low on that hierarchy. And it probably didn't have very strong reviewers, just as you're saying, Pira. It certainly didn't have reviewers who enforced compliance with the consort criteria, which is a very standard set of specifications of what data needs to show up in a report from a clinical trial. But if you read if this had gone to New England Journal, the New England Journal reviewers and editors would have enforced much higher standards on reporting. So anyhow, there's a broad spectrum. It's hard to know, and I don't know whether the clinicians can help guide the group here. But there's a broad spectrum of quality of journals out there that have different levels of review and editorial oversight.
So I'll just say two things. I mean, one, there is something called an impact factor that journals have, and you can find this on the internet. I believe the impact factor is the number of citations per page of the printed journal. So, you know, the higher the impact factor, the better the journal. So like New England Journal has a lot of citations for each page. I don't know what cytotherapy is, but I can't imagine it's very high. But even that's not perfect. And that's why we have this in the CRLI. I mean, I believe we live in the misinformation age, and it's very difficult to know who to trust. I think it's always best if we can empower people to investigate things themselves. And so now you are empowered consumers. You don't need to believe something that appears on Twitter or Facebook that says, hey, these guys in Mexico just found that stem cells work great for ALS. You can actually pull the paper up yourselves and read it, and maybe, you know, put a response that says, well, it's interesting, but, you know, here's some significant flaws in what they did that really limit how much we can believe the conclusion.
I don't know if you've shared it yesterday, Rick, but I've been, you know, touched by your story of the patients you've had who have gone to Mexico for stem cell therapy and declined what might have been, or certainly more proven therapies that were available that you were offering. But there's a real cost to misinformation.
Yeah. So we have a couple of comments, questions. Kevin writes, the progression rate of the treatment group was 0.77 per month, while the control group was almost double at 1.3. So certainly, Kevin, I agree that would look, oh my God, look at that. Except that the groups were different. And that's the problem. And those folks that were treated may well have been slower progressors to begin with. And so because of the design, you don't know. Matt says, where are good sources, journals to find these types of papers as they come out, specifically about ALS? Good question. You know, it's, you know, there are some sort of lay email publications like ALS Today that'll highlight important work, but of course that's not complete or thorough. You could go to PubMed and type in ALS once a month and see what came out and see if there's anything new. I don't know, Rick, do you have any idea where people can go, or, you know, to?
Yeah, Terry, I mean, one website that I have found to be generally very, very good, very timely, a very balanced, you know, view of different things that come out is ALS News Today.
Yeah, that's the publication that I was thinking of.
And then, I mean, I tried to keep ALS Untangled as balanced and updated as I can. I mean, we tend not to review things that are actually, you know, in trials because we are waiting for the trials. We know that's the best way, but there's a lot of other things, as you know, that are touted out there on the internet as ALS treatments without really any research behind them. And that's what ALS Untangled is all about, trying to uncover as many things as we can about the mechanism, the preclinical evidence, the case reports, the trials, and the risks. And again, it's just really to help people make more informed decisions about what they're gonna try for their ALS. It's not to shut anybody's clinic down or anything like that. It's purely to be educational for people with ALS who are trying to decide what they're gonna buy and try.
And ALS News Today comes out literally daily. You get an email. So it's something that everybody can sign up for. Gary,
sorry, Terry, I really want to applaud Rick, because I tell people all the time to really go to ALS Untangled, because it's a fantastic really service that you're doing to the community. Obviously, it is hard to keep up the pace, right? Because there is all that information there, but it is always incredibly good. It's a fantastic explanation of, is exactly the same exercise that we've been doing here today, which I think has an extremely high value for everybody. So I always tell people, go and check ALS Untangled or even I tell people, reach out to Dr. Bedlack and ask to do an ALS Untangled review on whatever you want to propose.
well, you know, it just shows Rick's commitment to PALS. between the CRLI that he's developed and ALS Untangled, which he's actually engaged the whole community in, all the academic neurologists who do ALS and anybody who wants to join in, we all look at the ALS Untangled. I don't know, Rick, what are we up to? How many ALS Untangled have you published now?
I think we're on 64, and I think we have 5 reviews right now in the queue, which means somebody has sent me a draft. And, between the first draft that I get and what you actually see published, there's a lot of steps. First there's my edits. And then, once I agree that the draft looks like it's something we can send out to the whole team, it goes out to 130 clinicians and scientists from across 11 countries. And they all get 2 weeks to weigh in. And sometimes they all weigh in and sometimes just a handful do. But their comments then have to be incorporated. And then, once that's done, we send it into the journal. It gets a peer review. And we have to incorporate the comments and edits of the peer from the journal. And then finally it gets published in the journal and on our website. So, the whole process, it takes about 40 hours of work from the time that somebody decides to do a review and the time that it actually gets published.
I would think it was even more than that, but and fortunately or unfortunately, there's so many more sort of non-traditional treatments to review. We can't possibly keep up with it, but Rick looks over the most commonly asked about, and that's what we kind of move towards. But Rick is to be applauded, and that, thank you, Piera, for pointing that out. Gary Curtis asks, since this was done in 2006, do we have an update? And Eric pointed out that there was a follow-up paper about safety, and the link is in the chat. If anybody's interested, you can go in the chat and click on it and see that follow-up paper. So thank you for putting up with this exercise, and I hope that you feel a little more comfortable reading, medical literature and how to approach it and get a start on it. Don't be afraid. You guys are all quite capable of reading something critically.
Terry, I would just go back to something I said yesterday too, and that is that we're available if you ever want to bounce anything off us. Like if you read something on the internet and then you read the paper yourself and you have questions about whether what's on the internet and what's in the paper really match, and you want somebody to take a look at it and see if they agree, I'm always happy to do that. I'm sure the other faculty here are always happy to provide that kind of bouncing off as well. I mean, I have to do that sometimes because that's what ALS Untangled is all about, is what I call crowdsourcing. So
honestly, I think I do that a couple times every week of my life, and it's my favorite part of the week, right? And you don't know what you don't know. And one of the great things about the ALS research community is that everybody has different wisdom, and I can reach out to other people if I don't know the answer. But I think this coming week, in fact, I've got calls scheduled with some people who are participating in this session to discuss exactly this, to talk about some results. So we work for you, and this is our job, and we love talking about this stuff. So please don't be shy.
I echo what was said. Please reach out to us anytime you have something. In terms of the stem cell research, if we go back to the questions of where the field is now, I think you all know that, Mayo Clinic has done mesenchymal stem cell research. The California Institute of Regenerative Medicine is also trying to advance. But what is very clear as kind of summary of those experiences is that at least the procedures in the US are safe, but as to efficacy, this has to be proven. So the field is moving, obviously, not at the speed we all need and want, but except for safety, we have no definitive data of efficacy.